Hi, I'm Dr. Spivak. I apologize that we are teaching or reaching out to you or communicating under these circumstances, but today I'm going to talk about clinical challenges in antibiotic resistance. And I would recommend that you listen to this lecture after listening to Antibiotics 101 and 102, which were in person recorded lectures that I put together. The objectives of today's talk are first to understand the history of antibiotic development, to identify major mechanisms of antibiotic action, to recognize mechanisms of antibiotic resistance once we understand the mechanisms of action and how those antibiotic resistance mechanisms are acquired. And I'm going to give some clinical examples as we go through the mechanisms. I'd like for you to be able to appreciate how antibiotic overuse and misuse leads to antibiotic resistance in the population and in patients individually, and the impacts that it has again on individual patients and society as a whole. You should be able to describe current efforts to minimize the spread of antibiotic resistant organisms and infections. Um, I apologize. And so those are the main objectives for this talk. So, the key concepts to start with overall that you should understand thinking about antibiotic resistance at a very high level, sort of a public health level for clinical care, is that although antibiotics have saved countless lives, and that is undoubtable, their use is not benign or risk neutral. There are individual consequences to a patient of adverse side effects or antibiotic resistance with overuse of antibiotics, and those have a societal cost as well. Antibiotic resistance is clearly a rapidly growing problem, and there are increasing rates of antibiotic resistance globally and nationally year to year. Paired with that, there are a few new antibiotics under development with pharmaceutical companies, and that is mainly due to economic issues and the fact that pharmaceutical companies, if they invest in the research and development for new antimicrobials, antibiotics, they are not often able to recuperate those costs in sales due to low volume need for their drugs, but also the recognition that folks should minimally use them or only use them as much as they need to, but not more, and so that limits their sales. Additionally, biologically, there are issues with if a bacteria develops resistance to an antibiotic, it is often very hard for us to develop or find new targets or new biologic targets to inhibit bacterial growth, and so that has limited the availability and the development of new antibiotics. Antibiotic resistance can be acquired by individual mutations in a bacteria, but is more commonly acquired by horizontal gene transfer. And we're going to talk about specifically what that means, but in a general sense, that means bacteria sharing their resistance with each other through sharing of genes, resistance genes from one bacteria to another. And those resistant antibiotic strains, if present in a bacterial population in a patient, can be selected for through the use of antibiotics that resistant bacteria may be resistant to, and then the resistant population can outgrow. Um, in general, and we're going to get to this, there are major antibiotic resistance mechanisms, three or four of them. One is to prevent access of the antibiotic to its target inside the bacteria. We'll talk about that. A second is to modify the antibiotic target, and we're going to talk about that, show schematics, and give clinical examples. And finally, modification or inactivation of antibiotics. And the classic example here is beta-lactamases that we'll talk about. So, antibiotic discovery. So, antibiotics in nature are synthesized by molds or bacteria, and these organisms synthesize and secrete antibiotics as a protective advantage to essentially inhibit bacteria in the environment around them and protect themselves. So, antibiotics were discovered by chance as the result of screening programs, most commonly. And nowadays, they can sort of be intentionally discovered by new molecular drug design. But the original antibiotic that most people are familiar with, penicillin, we'll talk about in a minute, was incidentally discovered from a mold, Penicillium, that was inhibiting the growth of bacteria surrounding it. Antibiotics are naturally occurring in nature, as we talked about, and these naturally occurring antibiotics can be chemically modified or engineered to improve their activities. So, penicillin was the original antibiotic or the classically remembered or touted antibiotic that was clinically discovered and developed. And then subsequently from penicillin, the aminopenicillins, ampicillin and amoxicillin, and the anti-staphylococcal penicillins and the anti-pseudomonal penicillins. These are all chemically modified versions of the original penicillin to enhance their spectrum of activity and their antibacterial properties. Prior to the discovery of antibiotics and vaccines in the late 1800s and early 1900s, pneumonia, tuberculosis, diarrhea, bacterial enteritis, and diphtheria, all of these essentially were the most common causes of death around the world and accounted for 1/3 of all deaths. So, to my point in the last slide, antibiotics have really been a significant discovery and advance in modern medicine, but we need to be careful in how we use them given development of antibiotic resistance and that may limit and jeopardize their usefulness. So, like I said, penicillin was one of the first antibiotics that was discovered. It was originally discovered by Alex Fleming at St. Mary's Hospital in London in 1928. And as you can see on the picture on the bottom, this is a petri dish that had Staph aureus colonies growing on it, and those are the small white dots. And as you can see toward the top, there's a much larger white colony, and that is penicillium, which is actually a mold or a fungus. And as you can see, surrounding and below that large colony, you see inhibition or less growth of these small white colonies. And that is because it was discovered that large mold, penicillium, was secreting a substance, later termed penicillin, in reference to the mold, that was inhibiting the growth of Staphylococcus aureus. So penicillin was rediscovered by Howard Florey about 10 years later after Alexander Fleming had discovered it. And later on, wide-scale production of penicillin happened in the early 1940s. And that manufacturing process was sort of discovered and optimized in the United States using new growth media and new molds. And penicillin was first then finally released for commercial use in 1943. Here on the right, you see just a picture to show you again sort of how amazing it was and the clinical response that was seen with initial uses of penicillin. This is pictures over time of a small child who had a group A strep facial infection. And these pictures here were taken in 1942, so she actually had access to penicillin. And you can see the improvement over time for this bacterial infection. So photo 3 here is 4 days after receiving penicillin, and photo 4 is 9 days, and then on the bottom 2 photos, she's fully recovered. So really, this was seen prior to penicillin. This child, unfortunately, would have probably died, but this was a dramatic response to penicillin in the face of a likely strep facial infection. So let's talk briefly, just as a refresher from, again, the Antibiotics 101 and 102 lectures about antibacterial mechanisms of action. This is a cartoon in the middle of a bacteria, so you can see in the teal out sort of ring around it, that is the bacterial cell wall. The pink is the periplasmic space, and the black sort of ring around it is the cell membrane. And inside, again, you can see the DNA and transcription and translation taking place on the ribosomes. And just to go around again, you can see all the different mechanisms of action or places or targets that antibiotics can have their effect on inhibiting bacterial replication. So as we've talked about, the greatest overall group are the cell wall synthesis inhibitors, most of them being beta-lactams, the penicillins, cephalosporins, monobactams, and carbapenems, but vancomycin, of course, is also a cell wall synthesis inhibitor through a different mechanism. And I won't go through all of these specifically, but the other main classes would be the protein synthesis inhibitors. That happens through varying mechanisms or targets, including different ribosomal 50S or 30S inhibitors and DNA-directed RNA polymerase inhibitors. We have the folic acid metabolism inhibition or inhibitors, trimethoprim and sulfamethoxazole, and the quinolones, which are DNA gyrase inhibitors. So I would encourage you to familiarize yourself with these main targets within the bacteria of different antibiotic classes. So, one thing that is commonly sort of a misconception is that antibiotic resistance is a newer process or a newer phenomenon that came about with the clinical use of antibiotics. But I'd pause for a second and ask you to think about this question: how old is antibiotic resistance? Is it something that came about in the 1990s? Is it prehistoric, maybe in the 1940s or the late 1800s? So the answer is that antibiotic resistance is prehistoric or essentially existed long before human use of antibiotics. And if you slow down and think for a minute, I told you that antibiotics, most of them are discovered as being present in nature. Penicillin, again, coming from a mold, penicillium, as a substance or a resource for it to inhibit bacterial growth and enhance its survival, the mold survival in nature. So it shouldn't be a surprise that antibiotic resistance is not a human phenomenon, but really just a natural phenomenon. These are two papers that very nicely illustrate that. The one on the top is from a group who sampled the microbiome or soil samples in an isolated cave in Carlsbad Caverns. And this is an area where no humans, not even rangers, had access. These researchers got access to this area and went in and took soil samples. And as I'll show in the next slide, they were able to find evidence of antibiotic resistance in many different bacteria that they isolated from this area. And this is an area, again, with no human contact, no animal or domesticated animal exposure, and no water runoff. Again, many of our antibiotics through being they gain water access essentially through the water supply from human and agricultural use. The bottom paper is another example from a different group, and this is a sampling of the permafrost in Alaska. They were able to date the permafrost based on genetic sequencing of animals that were alive during, and I forget what time it was, but essentially a prehistoric date. And they were able to find genetic sequences for well known antibiotic resistance genes that happen now in patients in clinical examples. And again, this is in area or dating the permafrost to a time before there was human exposure and human antibiotic use exposure as well. Again, making the point that the antibiotic resistance genes were there before humans were using antibiotics. This slide here is again from the first paper from the soil sampling in the cavern in Carlsbad Caverns. And what you're seeing on the top is gram-positive bacteria. On the bottom is gram-negative. Along the x-axis are various antibiotics, many of them antibiotics that we use clinically, and some of them non-clinical used antibiotics. And on the y-axis, you see the percent of these various gram-positive and gram-negative bacteria that were isolated from this cavern that expressed antibiotic resistance to the various bacteria. And the actual quantity is not important. The point being made is just that there is antibiotic resistance ranging from 10 or 20% to almost 80 or 90%, again, in an area where there has not been any human exposure, human use of antibiotic exposure, to point out that resistant bacteria exist in nature before we start using antibiotics. How antibiotic resistance, again, can happen if we recognize that antibiotic resistance is a natural phenomenon that then can be further selected for with antibiotic use and exposure between patients of antibiotic resistance genes. What this schematic then shows you is on the far left under number 1, if we say a patient has a bacterial infection, usually those infections can be with mixed populations. So some of the bacteria shown in blue are antibiotic susceptibles. The ones that are in deep pink can be antibiotic resistant. Or it could be that the patient has an infection with the blue bacteria, and the dark pink are their own normal bacteria that are not causing infection currently, but may have some resistance genes. As an antibiotic is given to this patient, The antibiotics kill bacteria causing the illness, so kill all the blue antibiotic susceptible bacteria, but they also can kill off some healthy bacteria. But what they then do is if they're killing off the susceptible bacteria, the antibiotics can select for the pink antibiotic resistant bacteria, and then those bacteria can then multiply, grow, and take over. And one thing again to point out, and I'll show this again later, is that antibiotic resistant bacteria can share their drug resistance. And this is again horizontal gene transfer. You may see it in some of these slides abbreviated as HGT, and share them with other bacteria, susceptible bacteria. So here a pink antibiotic resistant bacteria is sharing its resistance genes with a blue susceptible bacteria, which can cause a significant problem and make subsequent infections antibiotic resistant. So one of the main points for me trying to make the point that antibiotic resistance exists in nature and sort of predates human use of antibiotics is that since antibiotics have been discovered and used commercially in human healthcare and in clinical medicine, what we have seen is within a couple of years of every antibiotic that has been released for clinical use commercially, subsequent development of antibiotic resistance, which is here on the bottom of this timeline, within just a couple of years. So just to orient you again, this is just a real simple timeline with the year on the top that various antibiotics were deployed again for clinical use. The bottom is the year that antibiotic resistance was observed to each agent. And the specifics are not important, but just the point being made that very soon for most of these antibiotics, within a couple of years after there was deployment and use of these antibiotics commercially in large quantities, antibiotic resistance was detected. And again, the point being that shouldn't be surprising to us if we know that antibiotics were discovered incidentally in nature. The antibiotic resistance already occurs in nature because organisms are sort of developing resistance to protect themselves from each other. Then as humans start to use antibiotics, all we're doing is putting, pressure, large amounts of pressure on the gas pedal to accelerate the development or the recognition of antibiotic resistance. And so it shouldn't surprise us that we see resistance developing to these antibiotics sometimes very quickly. And I think how quickly it develops depends on essentially how much pressure we put on the gas pedal, which correlates with probably how much we're using the antibiotics. Alexander Fleming, when he discovered penicillin, and 20 years later, when it was released for commercial use, recognized this. He, in an op-ed piece in the New York Times in 1945, said, "The public will demand the drug, and then will begin an era of abuses. In such a case, the thoughtless person playing with penicillin treatment is morally responsible for the death of the man who finally succumbs to infection with a penicillin-resistant organism." So he had a recognition of the public health implications of misuse of antibiotics, and this is not a new issue. This has been known for quite some time. And again, we're now 75 years after he said this, and antibiotic resistance has really come one of the greatest challenges in medicine that Alexander Fleming really predicted. So, to give you an example of sort of some of the epidemiology of antibiotic resistance, this is one of CDC's maps. This is several years old, but they often track and will show state by state data of varying antibiotic resistant organisms of interest. And in 2001, we started to recognize that there were gram negative bacteria that had the potential to become carbapenem resistant. And if again you've listened to Antibiotics 101, you hopefully recognize that carbapenems, a type of beta lactam antibiotic, are one of the most broad spectrum antibiotics that we have to use that typically cover many, many gram negative organisms. And so in 2001, it was recognized that there were carbapenem resistant Enterobacteriaceae, or gram negative bacteria, in North Carolina. And as you can see here, this is the updated map 12 years later, showing significant spread across the entire United States. Subsequent data, again, this map is from 7 years ago. Now these organisms are detected in every state in the United States and in many places around the world, highlighting the spread of antibiotic resistance and how this can affect large geographic areas and many, many patients. In 2013, and this has subsequently been updated in 2019, the Centers for Disease Control, or CDC, released their first antibiotic resistance threats report in 2013. And this was the first time we had a real rough estimate. We do not have a national surveillance system, but this is data that is projected data based on the this limited surveillance we have through several public health departments and then applied to the national census. But this was the first look at giving us a sense of what are the numbers of antibiotic resistant infections in the United States and deaths annually. So they estimated that there are at least a little over 2 million antibiotic resistant illnesses in this country that result in 23,000 deaths. I will let you know that the numbers in 2019 that were updated are roughly the same, but with a slight increase. The estimated number of Clostridium difficile infections at this time was about 250,000. Updates to that look like around 400,000 C. diff related illnesses in the United States annually, with about 14 to 25,000 deaths. Clostridium difficile, if you have not learned about it yet, is a antibiotic resistant intestinal infection that a patient can acquire with healthcare exposure and antibiotic use. And it is a diarrheal disease that can be particularly disabling for a patient and cause large volume diarrhea. And in the elderly and comorbid patients with pre-existing conditions, can have some serious consequences and even lead to colectomy and death. Not only do antibiotics cause antibiotic resistance, but they also cause other side effects, as we've talked about in Antibiotics 101 and 102. It's been estimated that antibiotics are responsible for almost 1 in 5 emergency department visits for adverse drug events. They are the most common cause for emergency department visits for adverse drug events in children, given the large use of antibiotics in this population. And it's been projected by the WHO, if current trends continue, antibiotic resistant bacteria are expected to cause the premature deaths of 10 to 300 million people per year globally by 2050. So that's a bit of a very large overview of antibiotic resistance. And at the beginning, I told you one of the key principles was that new antibiotic agents are not being developed at a pace to keep up with the development of antibiotic resistance. And so this is just a bar graph to show you in chunks of 4 to 5 year periods how many new antibiotics were approved in the United States, FDA approved in those time frames. And as you can see, starting in 1983, the numbers per year were going down. And they went down pretty precipitously into the mid or early 2000s. In the last 7-8 years, there has been an uptick in development of new antibiotics due to some incentives and public and private partnerships. But many of these new antibiotics are for infections for which we do not need new antibiotics, and none of them are novel new mechanisms except for one. So the point being, there is some optimism, some increase in antibiotic development, but what we really need are new novel mechanisms to which bacteria are unlikely to develop resistance or not use resistance genes or mechanisms that they already have. I'd encourage you to go to this website at the bottom here, and this is a schematic in the middle of a it's called Faces of Antibiotic Resistance. It's a publication put out by IDSA, the Infectious Diseases Society of America, and this sort of 2-page spread here is a nice schematic and graphic just looking at sort of the history since 1943 of antibiotic use and antibiotic resistance. And this is meant to be a bacteria in the middle, and they're sort of going around from top to bottom, this bacteria, and really showing you the history of human antibiotic use and subsequent resistance. So on the top, you see on the top left, 1943 is the year that penicillin was commercially available for use. And really between the 1950 and up until the late 1980s, there was an abundance or a large increase in development of different antibiotics for clinical use. However, starting in the 1990s, between 1990 and 2000s, many and almost all of the pharmaceutical companies that were developing antibiotics got out of the antibiotics business, again, like I said before, because it was not financially viable for them. So that's when we really started to have the large reductions in FDA approvals and new antibiotics for clinical use. As you go around the bottom of this graphic, you start to see in the late 1990s and early 2000s that we see more and more antibiotic resistant infections. Start to see antibiotic resistant Neisseria, antibiotic resistant Acinetobacter and Pseudomonas, our carbapenemase producing infections like NDM1, which was first recognized in 2008, and onwards from there. I'll let you all read it in your own time. The point just being at sort of the top is the golden age of development of antibiotics, and then recognition after the 1990s and 2000s that antibiotic development has significantly decreased at the same time that antibiotic resistance is increasing. So let's switch gears now and sort of get away from more of the epidemiology and into the nuts and bolts of specifics of antibiotic resistant mechanisms. And I know you're going to get some of these in another lecture, but again, just to go through quickly some of the mechanisms with clinical examples. So as an overview, the main antibiotic resistant mechanisms are either to restrict an antibiotic access, so access into the bacteria, and this is either through porin mutations, or essentially mutations in the channels that antibiotics usually use to access the interior of the bacterium, or efflux pumps that essentially once the antibiotic gets in, the bacteria will just pump it back out, and those are represented in A. Another mechanism is modifying an antibiotic target. The target can be structurally altered so that the antibiotic can't bind to it, or the target can be overexpressed so that you would need much more antibiotic to inhibit the mechanism of that target. And that is represented in B on this graphic. Another antibiotic resistant mechanism is modification of the antibiotic itself. So usually inactivation before the antibiotic is able to affect its target. An example of this are beta-lactamases, which we will talk about. And then finally, another mechanism is modifying expression of bacterial factors needed to activate the antibiotics. And so this is usually in the form of prodrugs, where some antibiotics are given as a prodrug. We utilize bacterial enzymes that then go through a mechanism and or a change, and then will lead that we're sort of using bacterial enzymes to activate the drug to make it active against the bacteria. So let's start with the first mechanism, restricting access to an antibiotic target. Again, like I pointed out, this is either usually inhibiting uptake of the antibiotic through porin channel mutations, or boosting expression of efflux pumps, which will pump the antibiotic out. So a porin is represented. So this is a cross-sectional view of a gram-negative bacterial cell wall, and we see the outer membrane, the cell wall in the middle, and the inner membrane. So these are phospholipid membranes. The porins are usually on the outer membrane. These are these blue channels, and as you can see here, antibiotic A and antibiotic B can get through that porin, but because of the way it's maybe changed its conformation, antibiotic C cannot access through the porin. Once antibiotic A gets in, you can see it's binding to a penicillin binding protein, PBP, on the cell wall, and then it would inhibit its activity. Antibiotic B, the circle or spherical antibiotic, can get in through the porin, but as you see on the far right of the picture, there is a very large efflux pump that is essentially just pumping the antibiotic right back out of the bacteria. So these are two mechanisms of antibiotic resistance, and these are mechanisms we commonly see in gram-negative bacteria. Specifically, Pseudomonas usually has a lot of these mechanisms, or other gram-negative bacteria. So another mechanism of resistance is by modifying the target. So you've seen this picture before if you've listened to the Antibiotics 101 or 102 talk. So classic examples are penicillin resistance and vancomycin resistance. So let's orient to this picture here. So we are sort of inside the bacterial cell wall here. We have our transpeptidases, which are our penicillin binding proteins that cross-link peptidoglycan. These are the teal sort of non-teardrop shaped substances, I don't know what else to call them, in the middle of the picture here. Penicillin will usually bind to these. You can see the normal transpeptidase on the bottom toward the right. But if that transpeptidase mutates itself or alters itself so it has a little bump sticking out in the middle, then penicillin can't bind. And we'll talk about an example of an altered penicillin binding protein that leads to beta-lactam resistance. A separate type of or example of modifying the target is altered amino acid sequences where or altered amino acids, excuse me, where vancomycin would usually bind. Vancomycin usually binds to a diala amino acid sequence or amino acid, I'm sorry, on the inner cell wall and inhibits then these are peptidoglycan precursors and inhibits cross-linking of the peptidoglycan. If that amino acid chain changes, then vancomycin is no longer able to bind. So clinical examples of these, the most relevant is methicillin-resistant Staphylococcus aureus, or MRSA. Some people call it MRSA. Staphylococcus aureus, or gram-positive cocci. And this is an example, again, when we see methicillin resistance in Staph aureus of resistance by modifying a target. So what happens is by horizontal gene transfer, Staphylococcus aureus acquires what's called the MECA gene. This gene encodes for an altered penicillin binding protein, so an altered transpeptidase. That altered penicillin binding protein is abbreviated PBP2A. And what happens then is that methicillin or any of the anti-staphylococcal penicillins, so nafcillin, oxacillin, dicloxacillin, would not be able to bind to that penicillin binding protein and stop it from cross-linking peptidoglycan. So Staphylococcus aureus can be normal human colonizing bacteria living on mucosal surfaces, particularly in the anterior nares or anterior part of your nose, the nasopharynx, and the skin, usually around the axilla or the groin area. These bacteria can cause many types of infections, but usually skin infections, surgical site infections can get into the bloodstream through the skin and cause significant bloodstream infections with other distant site complications. The timeline on the bottom is just meant to show you again. So I think we talked about in Antibiotics 101 that Staphylococcus aureus originally were inhibited by penicillin. Then they started making a penicillinase. So again, that's a different mechanism. We'll talk about that soon. But an enzyme that modifies or inactivates the penicillin, and that was first detected in the mid 1940s. So then that's after penicillin resistance was detected in Staphylococcus aureus. That's when the penicillinase resistant or the anti-staphylococcal penicillins were developed. Again, methicillin was the original example, and it was introduced in the market near 1960. But then soon after that, methicillin resistance was detected. And again, this is the mechanism there is a modified target through alterations in a penicillin binding protein that methicillin can no longer bind to. It became then recognized in the mainly sort of from 1980 on that we started having increasing rates of methicillin-resistant Staph aureus that expanded and were well recognized in hospitals in the 1980s through the 2000s, and then sort of spilled over, we think, or developed maybe more commonly in the community as well in the 2000s. So let's talk about vancomycin resistance. Again, this is another example of antibiotic resistance by modifying the target. So vancomycin again inhibits cell wall synthesis by binding to the diala moiety, preventing cross-linking of the peptidoglycan. So resistance develops, we think, through a multi-step process. I will tell you that vancomycin resistance is actually fairly uncommon. It's one antibiotic we use. We use the antibiotic very commonly in the United States, but because resistance is a multi-step process, this is sort of the one clinical example I can think of where large volumes of antibiotic use don't seem to lead to significant resistance, but it is possible. So if you're going to have bacteria that's resistant to vancomycin, that usually that bacteria needs to acquire multiple enzymes via horizontal gene transfer. One is this VAN-H enzyme that catalyzes conversion of pyruvate to D-lactate. And then a second enzyme, it's usually either a VAN-A or a VAN-B that is acquired in a bacteria, again through horizontal gene transfer and uptake of genes from another resistant bacteria. That leads to formation of D-lactate. So instead of D-ala as the terminal sequence, D-lactate, and vancomycin cannot bind to that to inhibit cross-linking of peptidoglycan. Additionally, there is a third gene that we think that the bacteria acquires VAN-X, and this cleaves any existing D-ala formed by the usual pathway, thus preventing incorporation of any sort of other existing D-ala into the cell wall to sort of overcome the vancomycin resistance. So again, an example of another example of modification of the target in the bacteria. So vancomycin-resistant enterococcus can be a problem in healthcare. Enterococcus, just to refresh you, are gram-positive cocci that are usually in pairs or chains. They are normal human intestinal flora. But if they get out of the human intestines, they can cause several types of infections. Most commonly, they can get into the bloodstream and cause bacteremia. They can cause intra-abdominal infections if there's perforation of the intestines or the colon. And given their proximity to the urinary tract, can also cause urinary tract infections. Vancomycin-resistant enterococcus is often abbreviated VRE, and again, the resistance is due to modified target where vancomycin usually binds to inhibit cell wall synthesis. And again, I pointed out the main gene is thought to be the VAN-A gene that is plasmid associated and given to bacteria through horizontal gene transfer, and this is the gene that changes D-ala to D-ala D-lactate. Risk factors for VRE colonization infection we usually see this infection in patients who have had significant amounts of antibiotics exposure. Not that the antibiotic use is making it more likely that that patient has acquired the VAN-A gene, but we think that that antibiotic use selects for, so again, it would kill off healthy susceptible bacteria and allow the resistant organism to one, gain foothold in the patient's intestines and then proliferate as a significant bacteria in that niche in large quantities. So these infections tend to be in heavily healthcare exposed patients who have had a lot of antibiotic use. So let's switch and talk about antibiotic resistance by modification of the antibiotic itself. This usually happens. Most commonly, the clinical example is through beta-lactamases. So in this picture, you see here the beta-lactamase is the lighter blue sort of triangle shape. And that beta-lactamase has what it does is it hydrolyzes and chews up beta-lactam. Specifically in this example, it's penicillin. You see the sort of darker greenish grayish inactivated penicillin to the left. Then what that means is that the transpeptidase is able to just not be inhibited and go on cross-linking peptidoglycan and forming the bacterial cell wall. So again, just to show you sort of more with drawings here of the chemical structure. You can see the beta-lactam ring of penicillin on the left. Beta-lactamases again break a bond in the beta-lactam ring of penicillin and beta-lactams and disable the molecule. And bacteria with this enzyme can resist the effects of penicillin and other beta-lactam antibiotics. And we're not going to get into the weeds with beta-lactamases, but just to say there are different types. So there are penicillinases, enzymes that specifically only target penicillin. There are cephalosporinases. These are beta-lactamases that specifically target the cephalosporin family. And then there are more broad-spectrum beta-lactamases. What you can see here in this graph on the right, so just to orient you, time is on the x-axis. The y-axis is the number of viable bacteria. And so essentially this is meant to represent if we were to grow an amoxicillin resistant, so amoxicillin again, an aminopenicillin, we were to grow amoxicillin resistant E. coli in the presence of varying antibiotics. In the red, you could see if we grow it in the presence of amoxicillin, we see no inhibition in the growth of the bacteria. So over time, you see increasing quantities of the bacteria. Same if you grow it in the presence of clavulanic acid, which is another beta-lactam antibiotic. And there may be some inhibition of growth in comparison to control or no drug, but really the point being that over time we see increased growth of the bacteria. Now, when we put amoxicillin with clavulanic acid, which by itself is a weak beta-lactam, but is also a beta-lactamase inhibitor, what we see is that the clavulanic acid inhibits the beta-lactamase and then allows amoxicillin to do its job. So when you have the beta-lactam or the penicillin with the beta-lactamase inhibitor over time, we then see decreasing quantities of the amoxicillin-resistant E. coli. So this is an example of development of an antibiotic paired with a beta-lactamase inhibitor, making a new antibiotic, amoxicillin clavulanate or clavulanic acid, which overcomes that resistant mechanism of the beta-lactamase. A clinical example of beta-lactamases happened, there's numerous examples of them, but this is just one to show you a significant clinical results of beta-lactamase production. This is a report from CDC of a 70-year-old female who came to the U.S. She's a U.S. citizen, but came back to the U.S. in August of 2016 after she had had a prolonged visit in India. She had multiple hospitalizations while she was there due to a femur fracture, and she developed osteomyelitis or bone infection in that femur fracture. She, I believe, was from Nevada and came back to Nevada and was admitted in the hospital there in early August of 2016. She had been in the hospital multiple times in India and received multiple courses of antibiotics. When she came to the U.S., her wound culture grew a carbapenem-resistant Klebsiella pneumoniae. Klebsiella pneumoniae, again, being a enteric gram-negative rot or a member of the Enterobacteriaceae family, and carbapenem-resistant E stands for, or CRE stands for carbapenem-resistant Enterobacteriaceae. It was later identified that the mechanism of the carbapenem resistance was a what is now named a New Delhi metallo-beta-lactamase. So this is a beta-lactamase that is very broad spectrum and will hydrolyze or inactivate pretty much every beta-lactam, and most importantly, carbapenems, which are the most broad spectrum, are usually the most reliable against drug-resistant infections. Her Klebsiella pneumoniae was also found to be resistant to 26 other antibiotics, and unfortunately the patient died in early September of that same year. So the last sort of individual bacterial resistance mechanism we're going to talk about is resistance by modifying expression of bacterial factors needed to activate the antibiotic. So this is the example of prodrugs. Isoniazid is a good example of a antibiotic that is a prodrug and requires activation. So it is activated by the bacterial catalase or peroxidase enzyme CatG. Specifically, when we use isoniazid against Mycobacterium tuberculosis, that mycobacterial catalase peroxidase enzyme is what is used to convert isoniazid into an active drug. Isoniazid then inhibits the synthesis of mycolic acids, and these are the sort of backbone structure that are needed for the mycobacterial cell wall integrity. And so what you can see on the bottom here is on the far left, INH stands for isoniazid. The CatG is the enzyme that converts isoniazid into an active drug. And as we go through several steps there, then that drug would, or that active compound would inhibit mycolic acid synthesis. So if you have mutations in this first enzyme here, this CatG, you can never get conversion to active drug, and then you would have isoniazid-resistant Mycobacterium tuberculosis. So many of the resistance mechanisms I've talked about today, some examples of modification of the target, of enzymes that modify the antibiotic or inactivate the antibiotic itself. These resistance mechanisms are usually required by horizontal gene transfer. So it is possible for bacteria to develop resistance due to mutation, but as you see with the example with E. coli listed here, resistance by sort of individual mutation or by spontaneous mutation is fairly rare. So we think most of the resistance mechanisms that we see clinically are due to horizontal gene transfer, and specifically to plasmid transfer that you see here on the sort of top left of this cartoon under number 1, and this is conjugation. There is also the possibility for bacteria to take up free DNA from dead surrounding bacteria by transformation, or acquire antibiotic resistant genes by viral delivery, which is transduction. But clinically, we really think the majority of bacteria that we see acquire the resistance genes again by plasmid transfer. So transfer between bacteria of whole circles or loops of antibiotic resistance genes. And often these plasmids are quite large, so a plasmid that will have a beta lactamase on it may also have resistance genes to other antibiotic classes that are not beta lactams, such as fluoroquinolones, potentially folic acid synthesis inhibitors such as Bactrim, etc. So here's a cartoon example of this. It was on a shortcut through the hospital kitchens that Albert was first approached by a member of the antibiotic resistance. And he says, "Hey kid, want to be a superbug? Stick some of this into your genome. Even penicillin won't be able to harm you." So that's probably a gene or genetic material there to encode for a penicillinase or other beta lactamases that one bacteria is sharing with the other. Again, probably usually in the hospital, in healthcare exposures where we think this most commonly happens, although it can happen in the community. So let's switch gears for a minute and talk about antibiotic tolerance, which is not antibiotic resistance in an individual bacterium, but example of how colonies of bacteria can become tolerant of antibiotics. So bacteria that are normally susceptible to antibiotics may be able to tolerate these drugs, and essentially by tolerate I mean not be inhibited or affected by them under some conditions. Um, we think that antibiotic tolerance, so again, not with the antibiotics are not inhibiting the bacteria, contributes to a fair amount of chronic and recurrent relapsing infections that we see in patients. Antibiotic tolerance becomes important if we talk about bacteria that gain access to human cells and they have intracellular niches where maybe the antibiotics can't access them. If bacteria in their spore forms that are tolerant to heat, to cold, to acid, to disinfectants, they would also be tolerant to antibiotics, and that is a way for infections to persist. And most specifically, clinically, antibiotic tolerance we see when bacteria are within biofilms, which I'll talk about in a second, but are essentially large colonies or large ecosystems of bacteria that have developed mechanisms to tolerate antibiotics and not be suppressed by them. So bacteria present within biofilms are often, again, protected from antibiotics, and this is a schematic here of a biofilm formation where you see bacteria, which are these gold organisms adhering to a foreign body. Most commonly is when this happens in human infection. They then start to interact with each other. They have intracellular interaction and quorum sensing. They can induce this extracellular polysaccharide matrix that prevents antibiotics from accessing them. And they can sort of attract each other as well to grow their colony. So it's probably this polysaccharide capsule that prevents antibiotics from accessing this biofilm. And as we have more advances in modern medicine and we use more foreign bodies, prosthetic material examples would be cardiac pacemakers or orthopedic implants, prosthetic joints. It's these biofilms, it's this foreign material that bacteria have a predilection for forming a biofilm on, and then they become tolerant to antibiotics. And if this foreign material gets infected, it's difficult to treat them. Like I said, here is a clinical example of a biofilm infection or antibiotic tolerance. That is a cartoon of a prosthetic knee on the top right. Prosthetic joint infections are fairly uncommon. Less than 1% of all artificial hips and knees that are placed in this country get infected. But if they do get infected because it's foreign material and because of this biofilm possibility, they're very difficult to treat. So you can have an infection of a hip or a knee, arthroplasty. Arthroplasty is the official term for a joint replacement. And it's usually skin organisms, so most commonly gram-positive organisms that can inoculate the prostheses at the time of surgery or soon after if there's delayed wound healing. You can also sometimes get hematogenous or bloodborne seeding of the joint later on down the road, but most of these probably are acquired from skin bacteria getting into the joint. So small number of organisms can adhere to the implant, and then they start to form this protective biofilm that can evade antimicrobials and immunity. And this is an electron micrograph of a Staphylococcus epidermidis biofilm on foreign material that you see here on the bottom right. And these infections are very hard to treat. We do give these patients antibiotics, but the mainstay of treatment involves surgery and cleaning out the infection, and many times taking out the foreign material and placing it with new hardware, given the biofilm formation and the difficulty in clearing that off with antibiotics alone. So those are examples of antibiotic resistant mechanisms, and specifically clinical examples. So now we're going to change gears and talk about in clinical medicine and in the microbiology lab, how are we measuring antibiotic resistance and reporting that out? How is that measured clinically, and how does that what does that look like for a clinician? So this is an example from several years ago in our old electronic health record of what an antibiotic susceptibility report would look like. This is, as you can see in the blue, a report for an Enterococcus faecium. So that's the bacteria that was isolated from a patient's urinary tract. And what we usually get, as you can see here, a list of varying antibiotics from different classes. You see in the middle column in B, interp, and that stands for interpretation. And in the far right column in C, you see a number. And this is not formatted very well in this electronic health record, but on the far right, what that is the MIC, or the minimum inhibitory concentration, the lowest quantity in a lab setting, in a standard controlled setting of antibiotic that is needed to inhibit the growth of this bacteria. So the concentration of daptomycin that's needed, the concentration of linase load that is needed, etc. The interpretation we're going to talk about in a minute comes from or is directly related to what that minimum inhibitory concentration is, and essentially represents, do we consider an ampicillin minimum inhibitory concentration of greater than or equal to 16 as a susceptible or resistant infection, and it is considered a resistant infection, versus a concentration of 4 needed for daptomycin would be considered a susceptible infection. So that's what the interpretations mean. So how do we get at these minimum inhibitory concentrations, or MIC numbers? So the susceptibility testing, sort of behind the scenes, there's multiple methods that can be used. Most commonly, laboratories are using dilution methods or dilution tests. These can be the broth macro dilution or the automated broth micro dilution. I'll show you an example in a minute. And then there are also diffusion tests, which require growth of the bacteria on an agar plate and use of impregnated discs or strips with antibiotic to look at inhibition of antibiotic growth as the antibiotic diffuses out from that disc. And I'll show you examples of these as well. So the dilution test will actually give you a number, will give you a minimum inhibitory concentration, and the diffusion test will give you, we measure and you get a zone of inhibition, and then that zone of inhibition has been validated and correlates with an MIC for the organism. So here's a schematic of a broth macro dilution, and what you're looking at in A is susceptibility testing for an antibiotic susceptible organism, and in B, resistant organism. As you go from left to right, the test tubes have increasing concentration of antibiotic. That is the 0, 2, 4, 8, and 16. The concentrations are in micrograms per milliliter. Again, 0, so the first test tubes have no antibiotic in them. And again, we get increasing concentrations as we go from left to right. Each test tube would get a standard concentration of bacteria called a McFarland unit. And essentially, where the MIC is determined in a broth macro dilution, it's visual, and so the minimum inhibitory concentration needed to inhibit growth would be the first test tube in which there is no visual bacterial growth. So in the top example, it would be 8 micrograms per milliliter. In the bottom example, there's no evidence of inhibition of growth, so that's why this is an antibiotic resistant infection. A sort of picture of this. So this is a broth micro dilution. So this is the same mechanism that the broth macro dilution in the prior slide was showing you, except this is just done with on a micro scale. So with smaller quantities of bacteria and smaller quantities of antibiotics. But to just orient you here, sort of in each row would be, so this is I believe this was a Streptococcus viridans that is inoculated into each one of these wells. And on the top, you would see that as you go from so A is the row, but as you go from left to right, you would have increasing concentration in each one of these wells of clindamycin. And the sort of gold pellet that you can see is bacterial growth. So this would be interpreted. So in this far right well, there's 32 micrograms per milliliter of clindamycin, but we still see bacterial growth. So this would be reported out as an MIC of greater than, because we don't know what it is, 32 micrograms per milliliter. Some people have asked me, so each row would represent a different antibiotic, but in the example of row C, we would see that there is, and I don't know where the cutoff is, but one antibiotic probably put through this well right here or through well 7. And the MIC would probably for this particular antibiotic be at well 3. But as you go sorry, it's a hard time seeing the numbers. Well 8 is probably a start of a new antibiotic that is put there, and this is just how we're maximizing the amount of antibiotics that we can test in a 96 well plate. If people have questions about this, they can email me. So here are some pictures of diffusion tests. The top is diffusion, the bottom right is the E-test. And again, these usually involve inoculating a plate with whatever the bacterial isolate is of interest that's being tested. You grow it on the plate, and then you drop these, in the example on the top, these different discs. And each disc would have a different antibiotic impregnated into it. They are allowed to incubate for a certain amount of time and then read. And as you can see, some of the discs have a sort of clear zone around them where the bacterial growth is inhibited, and some don't. And there's a close-up at the top there of that as well. The zone of clearance around the disc is measured, and again, that measurement correlates with an MIC. On the E-test on the bottom right, this is a plate growing Staphylococcus aureus, and this is E-test strip. So this is a strip diffusion test that has increasing concentrations of vancomycin on it as we go from the bottom of the strip to the top. As you can see, with the higher concentration, and there's a larger zone of clearance around it. And the ellipse here, where the zone of clearance meets the E-test, is what is read out as the MIC. So this is another example of a diffusion test. So when I showed you the original microbiology report, we looked at the middle column, which was the interpretation. So many people ask, where does that come from? And it's kind of a confusing topic, admittedly. But there are what we call breakpoints or cutoffs for different bacterial and antibiotic combinations that determine that breakpoint is set for said bacteria and antibiotic combination as to whether or not that MIC is considered susceptible, intermediate, or resistant. So there are specific what we call bug drug breakpoints or MIC numbers that are set by various governing bodies. FDA is one group that sets these breakpoints for whether or not we would consider an MIC susceptible or resistant. And where the breakpoints are set is a process I've never been involved with, but is multifactorial and based on many things. So one thing that goes into that decision is if we took a whole population, let's say of samples from around the country of Staphylococcus aureus, what is the range or what is the distribution of MICs or minimum inhibitory concentrations of, let's say, vancomycin for that Staphylococcus aureus. Other things that go into the decision are pharmacokinetics, and also pharmacodynamics. So what's the attainable blood levels of said antibiotic that I can give? So, if we can never give a vancomycin concentration in a human to get to the minimum inhibitory concentration, then, that's something that goes into that. And pharmacokinetically, we can't achieve those concentrations, and the drug would be considered resistant. There's also evidence that goes into this, looking at the correlation of the minimum inhibitory concentrations and use of various antibiotics in clinical outcomes. Location of the infection matters. So sometimes there are different breakpoints for infection locations like the central nervous system or meningitis or CNS infections. Often it's hard to get antibiotics into the central nervous system. And so the minimum inhibitory concentration that's considered susceptible, often those breakpoints are lower for CNS infections because we have a harder time getting drug to that body site. So these are all the things that go into play in deciding what those breakpoints are, but your laboratory will usually interpret them for you and tell you either this antibiotic, either this bacteria antibiotic combination, we would consider this susceptible or resistant. So how does antibiotic resistance emerge in the real world? I've sort of given you examples or sort of talked epidemiologically or on an overview in the beginning of the lecture, showing you the timeline of antibiotics being used clinically or commercially and antibiotic resistance developing just a few years after that. But sometimes people don't understand like if I looked at one hospital or I looked at one clinic practice or a patient, what does that look like? What does overuse of antibiotics look like? So this is one study from CDC and some colleagues that they collaborated with in 2010 reviewing inpatient antibiotics from a subset of American hospitals. And they found, looking at these inpatient admissions, that over half of all inpatients in the United States receive an antibiotic at some point during their hospitalization. 30% of that is what we consider broad spectrum antibiotic use. So antibiotics, not like penicillin that covers a narrow spectrum of or a narrow group of antibiotics or only a few antibiotic, but broad spectrum antibiotic use that really will inhibit the growth of many, many bacteria. And what they found after doing chart reviews and looking at opportunities for improvement in antibiotic use is that up to 25% or almost a quarter of all this antibiotic use was potentially avoidable. So that is an example of saying, 25% of antibiotic use in the United States in hospitalized patients is potentially avoidable. That's a large scale evaluation. That's a significant amount of antibiotic use that could be improved. When we look at outpatient antibiotic use, so ambulatory, this is in clinics, mainly primary care clinics is where we usually look in the in the United States. We know that 80 to 90% of antibiotic use just quantitatively occurs in ambulatory settings. And that makes sense because only a minority of patients are in our hospitals. So most human antibiotic consumption is in the outpatient setting. And outpatient antibiotic use accounts for about 60% of the expenditures or money spent on all antibiotics in this country. 10 to 20% of outpatient visits, it varies if we're looking at children or adults, but 10 to 20% of outpatient visits result in an antibiotic prescription. We're just now beginning to understand these sort of new care delivery sites such as urgent care, and it looks like in urgent care 40 to 50% of those visits end with an antibiotic prescription. And when people have looked in the ambulatory settings, 30 to 50% of all of that antibiotic use in ambulatory or outpatient clinic settings is unnecessary or at least improvable. This is again, another map from CDC, and they put these out every year looking at community antibiotic prescriptions. Again, these are not in inpatient hospitals, but in clinic settings by 1,000 population by state. And what this is meant to show you is, I think it's hard to sort of quantitatively think about what is, 648 prescriptions per 1,000 population mean, but to just show you that there is geographic diversity and variation. In general, it looks like often the Midwest and the Southeast are higher prescribers, and the West is sort of on the lower prescribing spectrum, which is good. But having done a fair amount of this analysis and looking in Utah, I'll tell you, even though we look better, there are still significant opportunities for improvement because there is just so much overuse of antibiotics across the country as a whole. So how might an antibiotic be overused if we did one of those individual chart reviews and looked at an individual patient? In the ambulatory setting, it's fairly uncommon in how we can say that 30 to 50% of antibiotic use in the outpatient setting is unnecessary, is that it's very common for patients to get antibiotics for viral syndromes, for upper respiratory tract infections, for pharyngitis in adult, which is most commonly not due to group A streptococcus. And often in inpatient settings, we'll see patients getting antibiotics for non-infectious syndromes. So a patient may have pancreatitis or may have a septic type response that is due to an allergic reaction or to surgery that is non-infectious. Often we see people reacting to culture results. So Let's say a urine culture is sent for an unclear reason. Many people will have bacteria in their urine, but it doesn't correlate with infection. But that positive culture that represents colonization would be treated with an unnecessary antibiotic. Other examples of how antibiotics can be overused, people can often use overly broad spectrum. So again, let's say I have a group A strep infection, which are penicillin susceptible, and I give a patient a carbapenem. That's an example of overly broad spectrum antibiotic use. Using an antibiotic for an overly long course. So for community-acquired pneumonia, it looks like the vast majority of patients only need 5 days of antibiotics. If I gave a patient 10 days instead of 5 days, that would be an example of using an overly long course of antibiotics. Incorrect dosing is another way that antibiotics can be overused or misused. And then I'm not going to talk about it, but antimicrobial and specifically antibiotic use in um, the agricultural industry, specifically in animal feed, is a significant problem, and it accounts for probably more antibiotic use sort of in the United States than even human use does, and that can impact humans as we sort of share a geographic area with them, but also possibly through consumption of animal products. So, how are we going to improve all this antibiotic use and try and sort of rein in these problems and help people sort of prudently prescribe antibiotics? So, that is what I do at the University Hospital and the VA through something called antibiotic stewardship. So, there are guidelines around this, but antibiotic stewardship really is a multidisciplinary program, and we have programs at both hospitals, as does Intermountain, where our goals, and we work together as a team through various mechanisms and interventions. We try and optimize antibiotic use with the goal of improving patient clinical outcomes and minimizing the unintended consequences of antibiotic misuse. So we do things like review antibiotic courses on certain patients or patients in the hospital that are receiving certain antibiotics. We try and work with the lab and improve various diagnostic modalities that we have that would promote appropriate antibiotic use. We do a lot of education. We may restrict some antibiotics that we don't either need to be used commonly or have significant side effects. A whole host of varying things that we do to promote good antibiotic use. So some people say, what is antibiotic stewardship? And CDC sort of has what they call the 5 rights of antibiotic stewardship. It's it's using antibiotics in the right patient with the right drug at the right dose via the right route for the right duration. So the right patient being in a patient who has an infection, but specifically infection caused by a bacteria where the benefit of the antibiotic outweighs any risks. Right drug is picking the narrow spectrum drug. The right dose, picking the highest dose with the most favorable sort of benefit risk profile. So making sure we're giving the right amount of antibiotic. The right route is usually the least invasive and the most effective route. Many patients get intravenous or IV antibiotics when they could get oral antibiotics, which don't come with the sort of the downsides of having an IV with intravenous catheter-related side effects. And the right duration is always giving the shortest effective antibiotic duration to minimize unnecessary exposure. As I mentioned, the goals of antibiotic stewardship so these programs again usually involve physicians, usually ID physicians and ID trained pharmacists working together on this. Our goals are really to optimize patient outcomes and minimize unintended consequences. The unintended consequences can be antibiotic resistance, either in the short term or long term, but more importantly, we often see other adverse effects, whether it be rashes, renal toxicity, liver toxicity, diarrhea, or Clostridium difficile infections. These are all things that are on our radar that we're watching for to try and prevent with unnecessary antibiotic use. So antibiotic stewardship programs, I'm not going to go into the nuts and bolts, but it's been a sort of well-recognized quality improvement type initiative that hospitals and healthcare systems should develop. Our methods have been, there's not a great science behind them, but it's pretty clear about what types of interventions and monitoring you should do to improve antibiotic use. And now antibiotic stewardship is required in every hospital in the United States. So there's evidence to show that antibiotic stewardship can improve actually infection cure rates by guiding physicians on what the right antibiotics are to use for specific infections, can reduce Clostridium difficile infection rates, can improve patient safety again by trying to recognize and minimize drugs that are prone to cause adverse drug events and toxicities. We can reduce antibiotic resistance in the long term by minimizing unnecessary antibiotic use. Uh, Antibiotics do cost money, and so there are varying ways that we save money within the hospital setting, which we've done here at the University of Utah. And most importantly, it's a public health imperative. Hopefully, I've convinced you there's no question that antibiotic resistance is on the rise and has been for many years, and threatens a lot of the advances that we've made in modern medicine. We're able to do solid organ and hematopoietic stem cell transplants and give patients immunosuppressing therapy because historically we've been able to either prophylactically give them antibiotics to prevent infections or rescue them from their infection with antibiotic therapies. And antibiotic resistance really threatens our ability to keep these modern advances in medicine ongoing. So the pharmacist I work with at the VA has this great slide that he sort of tries to show this to this point. Why does antibiotic stewardship and how we use antibiotics matter? So again, antibiotics we need to think about as a shared resource that all humans are sharing. Most drugs, like if you think about medicines for blood pressure or chemotherapy for cancer, um, those drugs usually just affect one human or the target organ in that human that they're directed at. Antimicrobials, and specifically antibiotics, though, affect the human, so they kill off our healthy bacteria. They also affect the microorganisms that we are targeting or trying to target that may be causing an infection. But because they can induce antibiotic resistance, they really do affect other humans. So how you use antibiotics in one person really can affect the other person, can affect people in the community, people in the bed next to them, their family. And so it's something we want to think really hard about. So we're at the end here, and again, the objectives for this talk were to understand the history of antibiotic development, identify major mechanisms of antibiotic action, recognize mechanisms of antibiotic resistance and how they're acquired, and also identify those clinical examples I went through. Hopefully, you gained an appreciation of how antibiotic overuse and misuse can lead to antibiotic resistance and its impact on patients and society. And start to understand the current efforts. Antibiotic stewardship was the example I gave to minimize the spread of antibiotic resistant organisms and infections. So I'm going to end there. This is my email, and I realize we're all doing this remotely now. If you all have questions, feel free to email me. I am tied up in a bit of coronavirus stuff, but we'll get back to you as soon as I can. And I'm happy to answer any questions and hope everyone stays safe and healthy. Take care. Bye-bye.